I don't think people understand how much a single drop reveals. That little drop of dye can tell us a lot. From bacteria to tissues, stains speak loudly under microscopes. Let's decode them fast. First, what are stains? Simple stains color everything one uniform hue quickly. Differential stains separate groups based on chemical properties. Gram stain distinguishes bacteria by cell wall composition, purple or pink. Acid-fast stain highlights mycobacteria with waxy, lipid-rich walls. H&E, hematoxylin stains nuclei blue or purple. Eosin stains cytoplasm pink, creating classic tissue contrast. Quick demo. First, fix your specimen to preserve structure. Apply the primary stain for the correct time precisely. Rinse gently to remove excess without stripping signal. Then counterstain if needed to reveal additional structures. Dehydrate through grated alcohols to remove water. Clear with xylene or substitute to make tissue transparent. Mount with cover slip and mounting medium for microscopy. Tip: always run positive and negative controls alongside samples. Time your steps carefully. Consistency equals repeatable, reliable results. Overstaining can hide fine details and obscure structures. Understaining makes signals too weak to see clearly.